Hi, my name is Aline, and today I'll be talking about Streptococcus pneumoniae. This is a bacteria that causes serious illness around the world. Each year, in the United States, 1.3 million people visit their emergency hospitals with pneumonia. When this bacteria invades the lungs, it can cause pneumonia. When they occupy the bloodstream, they can cause sepsis, and when they affect the brain, they can cause meningitis. And there are serious diseases that can lead to death in more vulnerable groups like young children and older people. This is a prokaryote, which means is a simple cellular organism without nucleus and no membrane-bound organelles. And in the case of this bacteria, it is a gram-positive bacteria, which means that it has a thick peptidoglycan layer. It is a facultative and anaerobic bacteria, which means they can live with or without oxygen. And they're chemoorganotroph, which means that they rely on organic sources to make energy and grow. They also reproduce asexually by binary fission. Structure. It has a capsule, a cell wall that, like I mentioned before, is made of peptidoglycan. They have a cell membrane, inclusion body, nucleoid region, that's where its generic material is found. And we also have ribosome 70S, which means that they can make the protein synthesis. They also have pili. In this image, we see also how they can be found as a diplococci or a chain of cocci. Strep is for chain and coccus is for its round shape, so that's the name, Streptococcus. The life cycle and reproduction of this bacterium happens in our nasopharynx, and they can progress to be invasive, but most of the time we have a commensal relationship with them. During colonization, this microbe can be found extracellular, where they are able to encode the mucus that will entrap them and not allow them to progress to become invasive. They reproduce by binary fission, which results in two daughter cells. Binary fission is less complicated, and so it is faster in cell division in eukaryotes, contributing for them to grow and spread quickly. They have a circular chromosome with one orange of replication that extends in the copy in both directions simultaneously. And after binary fission had happened, they result in two daughter cells. And generic recombination of the generic material of Streptococcus pneumoniae happens by horizontal transfer. And in this bacteria specifically, they're able to do transformation, which means that they can take up DNA from the environment and incorporate in their own DNA, which makes them more pathogenic. Because since they're able to take DNA from the environment, they're able to become antibiotic resistant. Now we're going to learn a little bit about their ecological interactions. The first interaction that I'll be talking about will be how they interact with our human body, and the second interaction will be about the relationship with other microorganisms in our nasopharynx, and the third would be how they live in non-living surfaces and how they form biofilms. The interaction with the human environment happens because while they are living in our nasopharynx, they have a commensal relationship, like I said before, and they are chemoorganotroph, and they use as organic sources like carbohydrates. They are provided by the host to survive. In the nasopharynx, free sugars are usually scarce, but in the mucosa, the presence of glycans, they are able to use them as a source of energy. Still talking about the interaction with our human body, while they are in trying to be invasive, they're going to encounter immune defenses in the way. And the first line of defense is our mucus. The mucosa of the respiratory epithelial cells will provide a barrier against Streptococcus pneumonia. This mucus that entraps the bacterium, when it traps, they cannot access our epithelial cells, and this prevents infections. This mechanism is called mucociliary clearance. When trapped, the mucus in the respiratory cells activate other cells that release cytokines and chemokines. They also are able to kill Streptococcus pneumonia by releasing antimicrobial peptides such as defensine. How we can see this in this image, we can see that the bacteria tries to invade our cells. Here they're going to find antimicrobial peptides. The cells also is going to start releasing cytokine production to recruit immune cells. And we also see that the cilia is going to beat. That's how we call mucociliary clearance. Another interaction that they have, people were able to find that they form biofilms in non-living surfaces, and they can live there longer than was previously thought, and that's increased their chances of transmission. And they also have a relationship. Our nasopharynx is full of microorganisms, and they're trying to understand if this relationship can affect Streptococcus pneumonia. And scientists were found that there is an antagonist relationship between Corynebacterium accumbens and Streptococcus pneumonia. This happens because the Corynebacterium stimulates the host cell to deliver free fatty acids, and they are present in our nose sebum. Additionally, triglycerol lipase activity of Corynebacterium accumbens result in the release of free fatty acids and inhibits the growth of Streptococcus pneumonia. See, in this image we see the Corynebacterium accumbens is represented by this yellow, and we see how they release those fatty acids and how they inhibit the growth of Streptococcus pneumonia. In recent studies, we were able to find that the transmission of Streptococcus pneumonia also can happen by fomites. And in this experiment, scientists explored the survival of Streptococcus pneumonia under starvation conditions outside a host, and the role of its polysaccharide capsule in its survival. So in this experiment, results showed that the encapsulation allowed more prolonged survival than non-encapsulated bacteria. It was also noticed that the amount of encapsulation also affects the survival rate. The cells were incubated in a phosphate buffered solution at 25 Celsius. In the image, we can see 4 different types. T4 is represented by a full circle with no changing. It's expressing the total capsule polysaccharide. T4 PCAT is expressing only half of the capsule. T4 delta CPS does not express any capsule and is represented by the empty circle. And the T delta CPS that is represented by the square, it has its capsule restored. And each graph shows different results. The y-axis shows the survival percentage and the x-axis shows the incubation time in hours. After being in the solution, those cells were moved to a liquid and count down how much cells were dropped over time. And based on this graph, we can see that the strains T4 and T4 delta CPS that is represented. So we can see the circle and the square survive for the longest period of time compared to the other strains, which shows that the capsule does have an essential role in the survival of Streptococcus pneumonia during starvation. So the next questions could be, which kind of medications or products can be developed to target the capsule? How would this experiment work in humans? Does the size of the capsule matter in its survival? How does Streptococcus pneumoniae use capsules to prolong its survival? That's all about Streptococcus pneumoniae, and thank you.